Hello, good morning students. Today I would like to have the discussion of the next topic in plant histology and anatomy. We are discussing about the simple permanent tissues. In the earlier class we discussed about Sclerenchyma, then Sclerenchyma, how these two simple permanent tissues are different from one another, how they differ in their structure as well as in their functions. We discussed about that. Today let me consider another simple permanent tissue, that is Sclerenchyma. The term Sclerenchyma is derived from the Greek language. Scleros refers to hard. It is the hardest tissue present there in the parts. The most toughest tissue it is there in the plants. And it is said to be a dead mechanical tissue. As I told you in the previous class that mechanical tissues are the ones which are usually providing support to the plant. As far as Sclerenchyma is concerned, it is one of the simplest permanent tissues offering mechanical support. That is why Sclerenchyma was also referred as a mechanical tissue. And in addition to Sclerenchyma, there in the plants there is one more mechanical tissue present. That is none other than Sclerenchyma. This tissue, it provides support to the plant. It helps the plants to stand erect. It is not just the mechanical tissue. Remember, we refer it as a dead mechanical tissue. Because when you look into the cells of Sclerenchyma, assume that these are the cells that are present there in the Sclerenchyma. Cells of the Sclerenchyma, more or less, looks like this. What one could observe is, one could observe the presence of what you call as very thick cell wall present in the cells. Macrocopy, this is supposed to be the cell wall, which is very thick. Because of having the thick cell wall, this tissue is in a position to offer strength to the plant or support to the plant. That is why it acts as a mechanical tissue. When you look into the cells, what is there inside the cells? There inside the cells, there is an empty cavity present. Every cell is having the empty cavity. That empty cavity is considered as lumen. Means in these cells there is no protoplast present. Cells are without protoplast. When they don't have the protoplast, no living activities are taking place inside the cells. So we consider the cells as the dead cells. Let me consider how exactly these dead cells are formed and how is the structure of what you call as the Sclerenchyma cells and all. Very commonly, the cells present in the Sclerenchyma are usually called as fibers. We refer to them as fibers. Why do we refer them as Sclerenchyma fibers? or Sclerenchyma cells as fibers. When you look into the longitudinal section of Sclerenchyma tissue, suppose assume that this is a stem, a piece of a stem. Okay, let me say that here Sclerenchyma tissue is there inside the stem. This is supposed to be the Sclerenchyma. I want to look into the structure of the Sclerenchyma. How is its structure? That's why what I do is I am going to cut this stem along the longitudinal axis like this. When I used to cut this stem in the longitudinal axis, okay, I used to get the thin slices of the stem cut in longitudinal plane. These thin slices are usually called as longitudinal sections. We call them as longitudinal sections. Okay, in the longitudinal sections of the stem, you can see the Sclerenchyma tissue. And if you take one such longitudinal section of the stem and observe the Sclerenchyma tissue present there in the longitudinal section, how exactly the Sclerenchyma tissue appears? Okay, in longitudinal section, the Sclerenchyma cells more or less looks like this. Okay. These are supposed to be the Sclerenchyma cells. As far as the cells are concerned, they are very much elongated and they are looking like fibers. That is why we refer them as fibers. These are Sclerenchyma fibers. Every cell that is present there in Sclerenchyma fiber, it's usually having the thick cell wall. There is a thick cell wall present. Here am I copying. The cells are more or less looking like this. And these Sclerenchyma cells are very compactly arranged. They are compactly placed. Okay. And these cells, they don't leave any intercellular space. There are no intercellular spaces present. Okay. Suppose if you observe the spaces between the cells, absolutely you don't find the spaces because they are very tightly packed, they are very compactly placed. Every cell is having a thick cell wall. Okay. Cell wall is usually very thick. Even these cells, they don't have the protoplast, they have the empty cavity in the center. That empty cavity is usually considered as lumen. Because of absence of protoplast, we consider these cells as red cells. At maturity, Sclerenchyma cells are there. At maturity means when the Sclerenchyma cells are fully organized, fully produced. Okay, they become dead. They don't have any protoplast in them. They carry only the lumina, the empty cavity. Suppose if you observe a single Sclerenchyma fiber, the Sclerenchyma fiber more or less looks like this. This is how the fiber looks. Can you make out here? The cells are said to be broader in the middle and tapered at the ends. Can you make out? These cells, they are broader in the middle. This is approximately the middle region of the cell. Okay. If it is one cell, it is the approximate middle part. Here they are broader. When you move towards the poles or ends, yes, these cells are gradually pointed or tapered. Earlier, when the cells are produced at that time, okay, they are having the protoplast, means they contain the nucleus, cytoplasm, even the plasma membrane, everything they have. Okay. But what happens is, earlier, at the time of formation, these cells are having thin cell wall. The cell wall is usually thin. Can I make out here? This is the thin cell wall. This is the first formed wall. This first formed wall, which is usually the primary wall. This primary wall is made up of chemical compound that is cellulose. But what happens is, on this primary wall, lignin is going to be deposited. Lignin is the cell wall material. Okay. And this cell wall material is synthesized by the protoplasm and it is getting deposited on the primary wall. What happens is, once the lignin is getting deposited, with the deposition of that lignin, the cell wall becomes thick. Look here. This is the lignin. Once it is being continuously deposited, look here, it is being continuously deposited at all the places there on the primary wall. As a result of that, a thick uniform wall is going to be produced. And this wall layer that is produced because of lignin deposition is usually called as secondary wall, because it is later produced up to the primary wall formation. And this is produced due to lignin deposition, and it is made up of lignin. And this process of lignin deposition is usually called as lignification. And we refer Sclerenchyma as a lignified tissues. We call this as a lignified tissue because it contains lignin in the cell wall. Look here, primary wall and secondary wall together constructs cell wall. The together makes cell wall. And this cell wall is made up of chemical compounds like cellulose and lignin. Here cellulose is making the primary wall, lignin is making the secondary. Once the lignin is getting deposited, what happens is, here the plasmodesmata, the protoplasmic connections present between the cells are blocked. So that what happens is the protoplast disappears, nucleus, the cytoplasm. Even you also consider what you call as the plasma membrane, they all disappear. As a result of that, at maturity, then in the Sclerenchyma, you will find the presence of empty cavity. Empty cavity is none other than, look, we call it as loop. That is why we say at maturity, when the Sclerenchyma cell is being fully organized, it becomes dead. So we call this as a dead mechanical tissue. Even sometimes what happens is, when the lignin is getting deposited, at some places it is not going to get deposited. Look at this. Here, no deposition of the lignin. Maybe here there is no deposition of the lignin. Wherever lignin is not deposited, okay, in those areas you can observe the pit-like structures. These are supposed to be the pit-like structures. It is one of the simple pit that you can make out there in the cell wall of the Sclerenchyma fiber. And these pits are going to be produced or formed because of absence of lignin deposition. Okay, this is how the Sclerenchyma fibers appear in longitudinal section. Suppose if you observe the same Sclerenchyma tissue in the transverse section, how it appears? Assume that this is supposed to be the stem, okay, a piece of stem. Here Sclerenchyma tissue is present. If you cut the stem in the transverse plane, like this, transverse means exactly vertical to the longitudinal axis. Okay, if this is the longitudinal axis of the stem, if you cut the stem like this, it becomes what you call as the transverse section. If you take the transverse sections of the stem containing Sclerenchyma and observe this section under microscope, you will find the Sclerenchyma tissue more or less like this. Look at this. In transverse section, Sclerenchyma cell looks like this. First of all, you can see that cell wall are thick. Later I will come to that point. But here, shape of the cells. Look here, cells are usually polygonal in shape. They appear to contain many sides. When they contain more than one side, we consider the shape as polygonal. They are not only polygonal, even they are said to be compactly placed. Cells are compactly placed, and these cells are without any intracellular spaces. Look at the placement of the cells. Yes, cells are placed side by side, very compactly, without leaving any intracellular spaces. Okay, we call this kind of arrangement as a compact arrangement. Whenever there is a compact arrangement of the cells in C, we never find the intracellular spaces. Then these Sclerenchyma cells are usually possessing the thick cell. cell. And this thick cell wall is made up of cellulose as well as lignin. These are the two important chemical elements which are going to construct the cell wall. You don't find the presence of what you call as the protoplast here. You can see the loop. That's why we consider this tissue as a dead tissue, because cells are without protoplast. This is with reference to the structure of Sclerenchyma in the transverse section. Now let me consider the other type of Sclerenchyma cells. Usually the Sclerenchyma fibers. We consider Sclerenchyma fibers as most common type of Sclerenchyma cells. In addition to these Sclerenchyma fibers, there are also other Sclerenchyma cells present. These Sclerenchyma cells that are present other than fibers are usually called as sclerids. We call them as sclerids. In other words, these sclerids are also called as stone cells. They are as hard as stone. Wherever these sclerids are present, definitely the structures which are sclerids are like stones. They are as hard as stones. Say for example, usually in the seed covers. Usually in the seed covers, sclerids are present. Sometimes in the dry fruits, the wall is usually constructed with the sclerids. That is why they are quite hard. Why they are hard? What is the reason? The reason is repeating lignification. Repetitive lignification means the lignin deposition is not going to occur once. It is going to occur periodically, one after the other. There are many periodic intervals of lignification. Say for example, just consider that this is a sun. Say for example, this is a sun. Okay. In this cell, there is a primary wall that is being present. Okay, the cell wall is usually thin. What happens here is there is a deposition of the lignin occurs. Okay, when the lignin is getting deposited for the first time, okay, there is a formation of one layer of lignin. This is lignin that is going to be produced once. Over this, what happens is even the lignin is getting deposited for the second time also. Okay. When the lignin is getting deposited for the second time, another layer of lignin is going to be used. Like that, the lignification process never stops. It may occur for the third time, for the fourth time. Like this. When the lignin deposition is going to continue, what happens is concentric layers of the lignin are produced. Can you make out here? This is all nothing but what you call as cell wall. And this cell wall is very, very thick. Why it is thick? It is thick because of heavy lignin deposition. Once the heavy lignin deposition occurs and the cells become so much thick, cell wall, such thick cell wall cell, it can offer much strength to the plant and even toughness to the plant. Seed coats are normally very hard. because they contain what you call as the much thickened cells like slits. Usually in the slits, the lumen is very much narrow, very much obliterated. Why the lumen is very much obliterated? Why it is very less? It is very less because of repetitive deposition of the lignin. Continuous deposition of the lignin. Okay. All the slits that are present in different plant species may not be of the same type. They differ in the shapes. Based on the shapes, the slits are categorized into different types. Say here, we observe the slits of different types. The very first kind of slits are usually called as brachyslits. What are brachyslits? Okay, look at this. Brachyslits are like this. Okay, these slits are more or less spherical in shape. They are like this. And in these slits, one can observe the lignin deposition in the form of continuous layers. Here, concentric layers of the lignin deposition can be observed. Concentric layers means the layers which are formed one over the other. If I make out here, one could see the slits of this type, which are nearly spherical. Okay, we consider them as brachyslits. There are also second type of slits. We refer them as macroslits. Macro means big. Okay, these slits are elongated and more or less like this. They are like fingers. Okay. Even here also, you can observe a very much thick cell. Thick cell volcanics. Okay. We consider them as macroslits. Some kind of slits are there, which we refer them as astroslits. Aster means star. See, these slits are like this. They are looking like star. They have the radiating arms. Okay. Then they have a very thick cell wall. The cell wall is usually thick, just like this. They are looking like star. That is why we refer them as astroslits. Then comes osteoslits. Osteon means bone. Okay, these slits are like bones. They are looking like bones. Usually, the slits, they have the dilated edge, widened edge, and the narrow middle part. This is the narrow middle part. And these are the dilated ends or widened ends. Okay. looking at this, we refer them as osteoslithes. There are also some slithes that are present which are hair-like, and we refer them as trichoslithes. Tricho means hair-like. Trichoslithes are very much elongated. They are much thicker here. Sons, but only thing is they are very much elongated and looking like hairs. That is why we refer them as trichoslithes. Okay, these are the 5 kinds of slithes. Whatever may be the Sclerenchyma, maybe the fibers are the slithes. Because of their thick wall, they offer mechanical strength to the plant. Means the main function of this Sclerenchyma is support. It is a support giving tissue. It provides support to the plants. That is what we consider it as tissue. Okay, I would like to write up with this, and in the next class I am going to continue with the complex plant issues. Thank you.